It's 2025. Atmospheric concentrations of carbon dioxide are at 430 parts per million compared to the 350 parts per million maximum for a stable climate. As a society facing global climate change, we need to get to net zero greenhouse gas emissions by 2050. This requires a massive and rapid reduction in our greenhouse gas emissions, and those emissions that are still being emitted need to be sequestered. What are the solutions to do this?
As ecologists, we prefer to use what the planet already has, no? And it's what is called the natural-based solution. So it's a way of letting the planet to use its own immune system. So the planet has these ecosystems that capture and store carbon dioxide for centuries or millennia, and one of them are those coastal vegetated ecosystems, like seagrasses, salt marshes and mangroves. This is what we call the blue carbon. Basically, blue carbon is all the carbon that the oceans store, typically in their biomass and sediments. A healthy blue carbon ecosystem, a seagrass meadow, a mangrove, a salt marsh, they not only capture carbon dioxide, but they also, of course, provide many other ecosystem services, being biodiversity hotspots, protecting the coastlines from erosion, or capturing all kinds of pollutants. This storage has to be for more than 100 years, otherwise it's not too relevant for our purposes. And these ecosystems, they do retain the carbon dioxide for centuries or millennia. The most important service of these ecosystems is not the actual capture, annual capture of carbon dioxide, but to pay attention to the enormous amount of carbon that has been already stored over thousands of years. So we not only have to preserve them to keep the service of capture, but also to preserve the stocks that have been already laid down under these ecosystems. So any damage to seagrasses, salt marshes or mangroves, the danger of this damage is that enormous amounts of carbon dioxide can be re-emitted to the atmosphere. That's why conservation, rather than or in addition to restoration, are the two goals, the important goals to take into account. They are under threats. No? The main ones are the chemical pollution, eutropia, and above all, mechanical destruction. No? So it is important to preserve those ecosystems and above all the stocks that are being held by these ecosystems. How efficient they are in capturing carbon dioxide, we can only know that through research, through knowledge.
Our study site is Portlligat Bay, which is covered by 32 hectares of Posidonia oceanica. Our main study goals are to evaluate the changes of the Posidonia meadow cover across time and space, but also we want to quantify the carbon that is stored below. Ultimately, we want to create a 3D map where we can see the carbon stock, where it's stored, and which are the fluxes across time and across space. Also, we want to compare traditional coring techniques with new remote sensing techniques. Over the past few decades in the Mediterranean, we have lost 30% of seagrass meadow coverage. We want to see in Portlligat Bay if there's a regression in the Posidonia oceanic meadows, and why is that? So we will look at the factors that drive this recession. Was it environmental factors, anthropogenic factors? Those are the quick questions that we have.
So here at Portlligat, we are looking to break the bay into its many different types of blue carbon habitat. The main one being our Posidonia oceanica seagrass meadow. The meadow itself is structured into many different types. It's not just the living plant itself, but also the dead mat or the dead plant, as well as the sand and the other different types of gravel and mud that is based within the meadow itself. Each of these different habitats has very different characteristics of how the sediment is built, which influences how the carbon is stored within the environment itself. We are using primarily sediment cores, which are being drilled down into the sediment of all types, and then pulling out approximately 1 meter of sediment itself, which can tell us the entire life history of the meadow. We are using carbon-14, which is a technique to look at every single layer within the core itself to determine how old that layer is. By doing this and understanding what that layer is made of, we can then tell the history of the bay at that specific moment. In addition to this, we will be using many different types of analysis on the sediment itself. We will be looking at how the carbon, organic and inorganic, is structured. Sometimes it's packed very tightly together, like in mud, and other times, like in sand, it's more spread out and much looser. In addition to these methods, we'll be using sonar. The many different types include multi-beam, side scan, and sub-bottom, all of which just tell us different properties of what the benthos looks like, how the sediment is structured, as well as, without actually diving, what the environment is composed of. Multi-beam just tells us where the seafloor is. This method will show us where our seagrass meadow is located, how deep it can extend to, as well as the general characteristics of the area. We also use side scan sonar. Side scan sonar is taking a very high-tech picture of the seafloor. With this method, we just get the roughness or the texture of the seafloor. This will tell us if there is sand, if there is Posidonia, or if there is mud or some other buried material. Another type of sonar we use is known as sub-bottom profiling. This sonar allows us to look underneath the sediment and look at every single layer in detail of how the sediment and its properties are built on each other. Now with the sediment core, we can look at every single slice. When we use a sub-bottom profiler, instead we're looking at the differences. This means that we'll be looking at where the two sediment layers meet. This could be our dead mat, and this could be, for example, sand. The idea of the sub-bottom profiler is to then say, what is the volume? How can we directly, without taking a core, measure the entire buried carbon stock? Once we're able to measure a small area of carbon stock within Portlligat, we're then able to expand it across other similar habitats based on their various sediment properties. We are trying to find the volume of the mat of the blue carbon at Portlligat in order to show how valuable these ecosystems really are.
So one of the issues we face nowadays is the cost of restoring coastal ecosystems or marine ecosystems, because essentially going to the field and doing this restoration is very expensive. But we need to do it at a scale, because otherwise our actions doesn't have an impact on climate. So the research that we are conducting essentially provides very cost-effective tools to monitor fluxes of carbon, and therefore to really assess and robustly assess the benefits of our restoration in terms of carbon sequestration. In particular, these remote sensing techniques that we are implementing, they are very cost-effective and they can cover large, very large scales. So the emerging markets, including carbon, biodiversity, and other ecosystem services, provides a financial return for all these conservation and restoration actions. And we really need to capitalize on this opportunity. But essentially, if there is money to restore and there is investment and intention, then we should take the opportunity and make a difference by restoring ecosystems at scale.
So it's really important for us to monitor biodiversity, especially when we're doing seagrass restoration. So we want to know, when we've restored seagrass, have we recovered that biodiversity? Is that returning to the site? Because seagrasses are really important for marine biodiversity. They're the foundation of the food web. They're a nursery habitat and they support an abundance of marine life. And so one way that we're monitoring biodiversity recovery after restoration is using environmental DNA. And so environmental DNA is DNA that is shed by marine organisms into the environment. And this can exist in the water column or settle onto underlying sediments. And this kind of acts like a fingerprint of a marine organism. And what we can do is collect water, collect sediment, extract this DNA, and use that fingerprint to match to what organisms are present in the environment. And so a really interesting and novel way to assess biodiversity. It also means we can access areas that may be otherwise difficult to survey using traditional methods. And so we're developing this as a toolkit to monitor biodiversity recovery after restoration. But it also means that we can generate units of biodiversity from eDNA that we can then integrate into a crediting framework. So these biodiversity credits can be bought and sold, and this helps to fund continued restoration of seagrass meadows.
So to recap, these blue carbon ecosystems are incredibly important for biodiversity and they act as carbon sinks. These blue carbon ecosystems are nature-based solutions that will help us get to our net zero greenhouse gas goals. We need to implement this conducted research to help incentivise conservation and guide restoration efforts to keep the stored carbon in the sediments and to help incentivise drawing down more carbon to help stabilise our climate.